Okay, let's resume with aerobic training and adaptation. Okay, let us resume. We ended the other day talking about, let's stipulate if you train VO2 max goes up. Let's dig into now, what are the things that are going to change? All right, what are the things that are going to change? And so we ended on Wednesday saying that primarily there are no changes in anaerobic metabolism. Okay, and I mentioned that for the most part, the only enzyme in the Krebs cycle or that relates to the formation of lactic acid that changes appreciably is hexokinase. Okay, it's hexokinase. So PFK, there's a little bit of mixed evidence. Maybe if you train at super high intensities, you do interval training, maybe you can shift it around a little bit, but not a lot. There's really not a lot that goes on with LDH. You get a little bit of a decrease in type 2X fibers, but as we'll see later, the big thing that sort of blunts the formation of lactic acid is going to be what happens in the Krebs cycle and the electron transport chain, and that we rid ourselves of 2X fibers. We just turn our 2X's into 2A's, and so that's kind of the big deal that drives a lot of that. Okay, so the function of these adaptations, one of the big things, and this is going to really, in some ways, try to get at the hexokinase side of things, is increasing hexokinase lets us use more blood glucose, which leads to us sparing or not using muscle glycogen. Okay. Now I know we don't hormonal control. There's this push and pull between if we use blood glucose, that can lead to fatigue. We also know if we use up our muscle glycogen, that also leads to fatigue. And so there's a bit of a push and pull that's going to be there. But one of the things that we do see in people that are highly trained is they do spare their muscle glycogen. But most of that sparing is not from hexokinase, not from using less blood glucose. Most of it's from increased use of fat, which we'll talk about. Okay. Produce less lactate. We get a little bit of a down regulation in LDH because, as I said, the big reason we don't make as much lactate is going to be driven by Krebs cycle stuff and from the fiber type shift from X to A. But collectively, that's good, right? That's good. Less lactic acid, less change in pH, less fatigue, spare glycogen, less fatigue. Okay. All right. So you're going to go forward. So let's talk about what actually happens from an aerobic standpoint. Okay. What if I told you that in 8 weeks, I could double the amount of mitochondria in your muscle fibers? Double. Okay. If you have 100, now you have 200. If you had 200, now you have 400. Double the amount of mitochondria. In addition to having more, I make the ones you have bigger and better. Okay. These super jacked up, pumped up mitochondria. Okay. And that's indeed what happens. Mitochondrial content in type 1 fibers, type 2A fibers, and even in type X, 2X, if you have any, with consistent training, you go from having 1 to 2, and your 2 are bigger. Okay. And they're bigger. This is in conjunction with what happens in the cardiovascular system. This is arguably the largest or one of the largest effectors of your increase in VO2 max. It's also the thing that has a huge impact on metabolism at rest and metabolism during submaximal exercise. Okay. We're going to double our mitochondria. The reason this is such a powerful adaptation is that we go from having 1 set of Krebs cycle enzymes, 1 set of electron transport chain cytochromes and machinery, to now we have 2. You have twice as much. Okay. So now you can handle twice as much pyruvate going into the Krebs cycle than you could before. So it limits what goes and gets turned into lactic acid. You have twice the capacity to take those NAD and FAD molecules that have trapped electrons and donate them with the electron transport chain. So biochemically, this is one of the most powerful adaptations. Okay. All we've done is double our content. The enzymes don't work better, the enzymes don't work faster. Okay. You just have more PDH. Not a faster PDH enzyme to turn pyruvate into acetyl-CoA. You have more cytochromes, more ATP synthase. It doesn't work faster. It doesn't work any differently. There's just more. Okay? There's just more. And biochemically, this is a really, really powerful way for us to ensure that all, or not all, but way more pyruvate gets shoved over into the Krebs cycle, and we don't have to turn it into lactic acid. Okay? So we are in essence doubling our capacity to turn pyruvate into ATP and to turn free fatty acids into ATP. Okay? Very, very, very powerful adaptation. So some really, really kind of old data where we began to see this. This is some of the earliest biochemical evidence of this from one of the most famous. I think I've shown you guys data from him before. One of the most famous exercise physiologists ever. His name is John Palazzi. He was at Washington University in St. Louis and was one of the first people to ever actually run on a treadmill, rats and mice, and check out what's happening in their muscles. Okay? You don't want to know how you run a rat and a mouse on a treadmill, right? It's not great. It's really not. They put a shock thing at the back of the treadmill, and if the rat and the mouse doesn't want to run, it shocks them, and then they run. Okay? You're literally chasing them like a cattle prod on all of that. Okay? So what you see though, and I just want to highlight this, right? In the top panel, you've got gastroc and you've got soleus, you have sedentary rats or mice, and then exercise. I think this was in rats. Sedentary rats that exercised on the treadmill, right, for 12 weeks, 5 days a week, 10 to 30-minute bouts. They progressively increase the speed and the grade on the little rat treadmill, okay? And you can see cytochrome oxidase and succinate oxidase. So this is a, right, that's a enzymes in the Krebs cycle. Then I want to look at this. They actually looked at mitochondrial protein content. So you've got more of these enzymes because there's more mitochondrial content. Okay. And so it went from about 3 milligrams per gram of muscle to about 4 2/3. Okay. So not quite a doubling in these. In the human data, we can see this is just massive, massive increase. Okay. Massive increase. These rats go from being able to run for like 20 minutes to be able to run for an hour at a higher incline and much faster speed. Get these very, very massive increases in aerobic metabolism. Okay. So this may raise a question for you guys, which is, Dr. Black, how do I get more mitochondria? What's the signal? Why does training do this? Does some kind of training work better than others? Okay. And the short answer is almost all aerobic training, if the intensity is sufficient, will do this. If you want more mitochondria, you have to tax the mitochondria that you have. You have to make the ones you have work hard to make more ATP. Okay. So what we can see is when you train and you drive mitochondrial ATP production, it activates, okay, one sort of intracellular signaling molecule that I want you guys to know. This is the mTOR, or the mTOR analog of aerobic training adaptations or mitochondrial adaptations. And it's called PGC1 alpha, which stands for peroxisome proliferator activated receptor gamma coactivator. Right? It's just, we just call it PGC1 alpha. And it works like mTOR. When you make mitochondria work hard, it turns on the genes and you get more PGC1 alpha. And PGC1 alpha activates the other genes in the mitochondria. Mitochondria have their own set of DNA, their own set of genes. And it tells them to make more mitochondria. Okay? This is the kind of central hub. And so what you can see basically is pre-training, post-training, we dramatically increase PGC1 alpha content. Okay. On the right, you can see some data where you're looking at pre-training and post-training in type 1 fibers, type 2A and type 2X fibers, and you'll note it goes up in all of them, and it really goes up in 2A fibers. Okay. Your type 1 fibers have a lot of mitochondria to begin with, they're going to get more. Your 2A fibers, if you're sedentary, don't have that many mitochondria, and it dramatically increases. Dramatically increases. Just to see what happens, you're not responsible for knowing anything on this slide, other than here's PGC1 alpha, and it has all of these different signal effectors down here. MEF1, NRF1 and 2, PAR alpha, and those things, all of which generally lead to the activation of the mitochondrial genes to make more mitochondria. And what you can see is the thing that activates PGC1 alpha is something called AMPK or AMP kinase, which gets activated by energy deprivation. You exercise, you force the mitochondria to work hard. That activates AMP kinase. AMP kinase also plays a role in resistance training adaptation, but you're using lots of energy, it turns on PGC1 alpha. That's the big thing. Okay. So when I tell you that jogging for an hour at 60% of VO2 max does this, and you get the same response if you do 5 high-intensity 30-second intervals with about 2 minutes of rest in between. What you have to imagine is at a lower intensity, it takes a longer amount of time to activate AMP kinase to get that energy deprivation, right? Because you're not using as much every minute. When you do super high intensity stuff, you know, a lot of energy deprivation, you make those mitochondria work really, really hard. And we know that we see similar adaptations to high intensity interval training compared to kind of traditional, kind of nice, easy jog, the same number of days per week and all. Okay. So saying all that, anybody want to meet me? Meet me at the Huff today. We'll just do sprints around the track. Do 1 lap on the track, and we'll walk a lap. We'll sprint a lap. We'll do that 5 times. Okay. Or we can go over to couch and we'll sprint up the stairs in the dorm. There's actually was a study, the folks that do a lot of interval training research at McGill University in Canada, they did a study, they did 20-second sprints up the stairs in their dorms. 3 20-second sprints up the stairs. Okay, then you take 40 seconds off and you do another. Do it 3 times. And they get a roughly similar increase in VO2 max as you get from running or biking at like 60% of VO2 max for half an hour, 4 or 5 days per week over 8 to 12 weeks. If you sprint and you just blow it out, it really drives mitochondria. That really, really high energy use is really important. Okay. Interval training works, it just sucks. Right? It just sucks. I think I've asked this before in either testing a prescription or the exercise physiology lab class. You guys do a Wingate test? Anybody seen a Wingate test? Yeah. So you've seen one? Did you have to do it yourself? No. What did it look like? Can you describe it for the class? It's very simple. You remember how long it was?
No.
So, okay. A Wingate test. You're on a bike. We give you a resistance that's 80% of your body weight. And we tell you to pedal as fast as you can for 30 seconds. all out sprint for 30 seconds. Okay? Do that. It's the most terrible thing you've ever done in your life. I don't care what you think you've done that's bad, this is worse. Okay? Do that. Do your 30 seconds, take 2.5 minutes off, just pedal on the bike. Do it again. Do that 5 times. 3 times per week. You've done 2.5 minutes of exercise. 2.5 minutes in a bout. And you get the exact same adaptations that you get if you run for a half an hour, 45 minutes for those same number of days per week. But it's terrible. Nobody wants to do it. It's absolutely terrible. So, but it works because both things lead to a similar level of energy deprivation and activate PGC1. Okay, that's for mitochondria. The cardiovascular side of things, it's a little bit different. But mitochondria, that's what we want to do. Okay, so we talked about this. How does having more mitochondria limit fatigue? It's biochemistry. Okay, it's biochemistry. If I tell you guys, right, that we're in this room, and I know it's Friday, not everybody is here, but if we had a full complement of all 80 of y'all here, and you close your eyes and you walk around until you bump into somebody else, right? If there's twice as many of you in here, you have twice the chance of bumping into someone. Biochemically, we call that an increase in sensitivity. There's a greater likelihood that a reaction will take place. So when you have more mitochondria, there's a greater chance that any pyruvate molecule will bump into a PDH enzyme and will get converted into acetyl-CoA and go into the Krebs cycle. Okay? There's twice the chance. That's good, right? We're increasing our biochemical chances of that happening. So when we do this, more pyruvate can go. The other side is there's now also twice the chance that any molecule of free fatty acid that gets into a cell can go into the Krebs cycle as well. One of the biggest adaptations to aerobic training than having more mitochondria is people that are trained derive a greater percentage of their caloric expenditure from fat at all times. Okay? You want to be able to eat more Doritos and Chick-fil-A, go run. You'll metabolize more fat at rest. Okay? More fat at rest. You want to metabolize more fat at any given exercise intensity that's below max. Have more mitochondria. Because the system is more sensitive to the presence of fat because there's a greater likelihood that reaction will take place. Okay? So, we're not going to talk about it in class, I probably should, but the argument for what we call concurrent training, where you do resistance training and you do aerobic training, you resistance train and you increase muscle mass. So you have more muscle, you can more than double your mitochondria because now you have total, because now you have more muscle in which you can double the mitochondria. You have more muscle, so your resting metabolic rate goes up. And you're better trained, so you have a higher metabolic rate and it metabolizes more fat. That in some ways is the way to sort of supercharge your ability to eat Chick-fil-A, what I say, or eat Doritos, or whatever your sort of lovely high-fat American diet food of choice is going to be. Ice cream, whatever it is.
We talk about how like muscles and like the size and the strength of them are like reversible. Like keep training, I guess, but if you don't train for a long time, you can really train. It's kind of all that.
Absolutely. We will get to detraining at the very end of this, but yes. Just like when you stop training, your muscles shrink. When you stop training, the mitochondria go away. Okay? You'll see we're going to get an increase in capillaries. They will go away. The hypertrophy in your left ventricle doesn't go away quite as much. Okay, but the mitochondria will go away. They come back pretty quickly. Your mitochondria are always getting turned over, kind of like your red blood cells. Right? They don't exist into perpetuity. They're there, you use them for a while, they get a little bit of damage, they get broken down, and then you make some new ones. And so without the stimulus, there's a kind of this natural time course of them going away. And it's, I would have to look up what it is, but they will go away if you stop. Okay. I'm going to put this here, the next adaptation, as a muscle metabolic adaptation because that's where it sort of manifests itself, is it helps us with metabolism. But it's actually a cardiovascular structure that adapts, okay? Having more mitochondria doesn't help us if I can't get more substrate and more oxygen to those mitochondria, right? Having a larger, right, having a larger cup of water doesn't do me any good if I'm drinking from the same little bitty straw, right? If I get a bigger straw, then I can get more of that larger, that larger tank of water. So the other adaptation that helps us be able to use more carbs in the Krebs cycle, more fat in the Krebs cycle, which requires more oxygen, is an increase in capillaries. When you train, the muscles that have been used during exercise will begin to have more and more capillaries around those muscle fibers. We call that capillary density. Number of capillaries per fiber. Okay. This is another very powerful adaptation. So what this does then is it allows us to direct, when we open those arterioles, to direct more fat, more glucose, more oxygen to those fibers that now have a higher mitochondrial content. Okay, so we get more blood flow there. That decreases the distance that those molecules have to diffuse to get into the fibers. Okay, that's going to be a positive. It also slows down blood flow. It also slows down blood flow. Having more places for the blood to go in those capillaries, it slows down how long it's going to progress. Okay. And if you imagine that the blood moving past the muscle fiber is like you guys having like an assembly line in front of you where stuff is sort of sliding along in front of you, and you need to grab stuff off or put stuff back onto that little assembly line. And the slower that it moves, the easier it is to pull stuff in and then put the CO2 and stuff back. So it does a really, really good job of that. It also increases our ability when we do make lactic acid to remove that lactate. It increases our capacity to shuttle that lactate sequence. So you need both capillaries and mitochondria to affect the maximum kind of change. So of the components of the thick equation, we'll critical think here. Having more mitochondria and more capillaries alters which component of the thick equation, which would therefore predictably make VO2 max go up. VO2 difference, that's right. Very good. This allows us to increase oxygen extraction. It makes VO2 difference go up. And the O2 extraction goes up because we're using more oxygen in those greater numbers of mitochondria. Very, very powerful adaptation. There's going to be another one that we're going to use in conjunction with these two things. But this is going to be a big deal. Okay. There is some debate about whether slower, kind of more traditional aerobic training does better from an increasing capillary density standpoint versus interval training. And I don't have a good answer for you guys. We know that both of them will make capillaries go up. Some studies say it's basically the same. There is some data that maybe longer, kind of traditional, slower distance training, maybe it's a little bit more. I don't know if we've settled that answer. Okay. But both are going to make VO2 difference go up. We need them both. Okay. Okay. Now, this next one's going to be a little conceptually difficult for you guys. Okay. And so this is an adaptation, and that's a correct way to frame it. But what it really is it's a change in a physiological measure that is the result of the changes that we just talked about. Okay. It's a result of having more mitochondria. It's a result of having more capillaries. And that is that if the exercise intensity at which you cross over lactate threshold is going to rise. Okay? And what typically happens is two things, okay? If you are sedentary and your lactate threshold occurs at 60% of your VO2 max, and let's say running at 6 miles an hour on a treadmill gets me to 60% of my VO2 max, okay? What generally happens is a combination of two things. If I make my VO2 max go up, okay? But lactate threshold as an intensity doesn't change as a percentage. My max goes up. So now 60% of my max occurs when I run at 7 miles. Okay. I haven't shifted lactate threshold as a percentage of max, but I can now run faster and before I get to my threshold. Does that make sense, guys? Okay. Max has gone up, the percentage at which the marker. Is there a marker? No, of course not. You see. Anybody have a dry erase marker? It'd be great. I can check for all this. If you got to just throw it at me. You think it will work, okay? And some of y'all.
Firefoot.
Got it. Okay. Okay. You guys see it this way, okay? Lactate threshold is at 60% of my max. Okay? And if we've got speed over here, like how fast I have to run, let's say this is 6 miles an hour, and then my max occurs at 8 miles an hour. Okay? After training, two things can happen. Okay? One is that max gets bigger, but lactate threshold is still at 6. I didn't draw that very well, but it's still at 60% of max. Okay. But what happens is that now, basically, I can run at 8 miles an hour before I get to lactate threshold. Okay. So you've increased the pace at which you can run before you start making trip over lactate threshold. The other thing, the thing that most commonly happens is not this, but that max goes up and then where lactate threshold occurs as a percentage of your max, we'll say now it's at 85% of max also goes up. So you're going to get this kind of double impact of that my threshold as a percentage of max has risen, and right, because my max has gone up, both of those things are now going to allow me to work at a higher rate. Okay, this comes from our ability to have more mitochondria and more capillaries, and therefore make less lactate. Okay, we make less lactate at any given work rate because we're able to use more fat, we're able to put more pyruvate into the Krebs cycle. Okay, that's because of increases in mitochondria and capillaries. It's also mildly due to a decrease in epinephrine and norepinephrine response to exercise. Okay, we get a blunted epinephrine and norepinephrine response as you become more trained. It's less stressful to you. Okay. We also get an increase in our ability because of more capillaries, as we will see a larger blood volume, a larger cardiac output. We will have a larger ability to remove lactate from the muscles and send it someplace else to either be buffered or to be shuttled and used as energy. Okay. So there's this kind of combination that we make less, and even when we do make some, we can do something else with it. Okay. What this functionally does for you is it allows you to run faster and feel the same without it feeling like you're over lactate threshold. Right. Now your actual jogging pace or your race pace or something can increase And you can maintain that faster pace kind of indefinitely because you're not making a lot of line. Okay? As an actual performance metric, this is one of the biggest things. Okay, that's going to happen. Okay. The last kind of metabolic adaptation, as I talked about, again, it's not a purely metabolic adaptation, it's really a structural thing, but it's related to metabolism is there's an increase in 2A fibers, we turn our type 2X fibers into type 2A. Okay, 2X to 2X. And then those 2A's get a whole shit ton of mitochondria. That's the usefulness of that. Okay. Why would that be important? Again, right, we're shifting the myosin ATPase efficiency, we're shifting the LDH concentration, and we're increasing mitochondria. So that's having kind of a big powerful effect. All right. So I already mentioned this, so I'll just kind of put it up here and be like, see, having those metabolic changes, as I talked about, it results in a shift in substrate utilization. So you're going to use more fat and less carbs at any intensity up until max. Okay. And for the carbs that you do use, we're going to spare glycogen, especially muscle glycogen. And that's going to help us then from a fatigue standpoint. Okay, we're going to maintain blood glucose better. We're going to spare muscle glycogen better. And that's going to allow us to go for a longer period of time, whatever our work rate is, because we're using more fat. Okay. So at any given work rate, VO2 or the oxygen needed doesn't change very much unless you change your weight or your running kind of mechanics. Okay, because you weigh the same, you're running at a certain speed, requires the same amount of force, same amount of energy, but our RER goes down and we make less light. Okay? That's one of the big things. Having mitochondria, okay, lets us shift our substrate utilization, and that has very powerful effects on fatigue. Okay, we talked about this already. We talked about the increase of fat use. Okay, why do these increase performance? Well, spares glycogen, right? Because of increased fat use, that helps us in long duration exercise. Weirdly, we're going to get into this a little bit later. One of the other things that happens during short duration exercise, and we don't know for sure how much of this is psychological and how much of it is physiological. I would love to do a study if anybody wants to come and do wind gate tests in the lab with people to train and be very comfortable. Is we know that people can tolerate greater changes in pH, greater acidity from an increase in lactate production. We get a little bit of an ability where you can better buffer hydrogen ions, okay, as you become more trained. But the other issue is that you may just tolerate pain and discomfort better because you're used to it from your training. Okay, there's an idea. This is a great segue. Y'all are going to get an email from me today about a survey that one of your fellow undergrads, a McNair scholar, is doing in my lab for her McNair project. And it's going to ask you questions about a concept called catastrophizing. Anybody know what it means to be a catastrophizer? Any of you that have an anxiety disorder are very likely a catastrophizer. Okay. A catastrophizer is you always think about the worst case scenario, right? Something happens. This hurts. Oh shit, bad things are about to happen. You catastrophize. Okay? Very common. We know that people that have chronic pain conditions are more catastrophizers than others. Okay? We know that in general, well-trained athletes are lower in their catastrophizing to pain than others. We don't know if that's a genetic thing or if it's a response to training. But the idea is you train, it hurts, and then you figure out that nothing bad actually happens. You stop, it goes away. The world did not end, right? It was not so bad. You forget how bad it was, right? And then you go again. Okay? And we know that there's some evidence that people that train, doing interval training where it hurts really bad, will see a greater ability to tolerate pain. And that may lead to increased performance in kind of those sort of somewhat very, very highly anaerobic kind of performance conditions, like a 400-meter runner. Things are gonna last, from like 30 seconds to about a minute and a half or two. There's some really interesting stuff. You're gonna get a survey. Please take a look at the survey. Okay, it's completely anonymous. It's gonna ask about catastrophizing. It's gonna ask about childhood trauma, which I'm very interested in. And it's gonna ask about a thing called stoicism. You guys know what stoicism is? Yeah, what's stoicism? When it was not in your head.
Like you're kind of you're like stoic, like you're not.
It is stoic. What does it mean to be? You guys know what does it mean to be stoic? I mean, it's been a while since you've had like intro to psych, or something, right?
Like the desire to follow the rules, or like be strict on things.
Maybe kind of sort of, you're kind of hitting around it. Who do we generally think of as being stoic? You guys,
Boring.
boring people? Okay. Not exactly. Y'all are in some ways kind of a generation removed from something.
Like headstrong,
Headstrong, we're kind of all, those are things that might be there, right? Stoicism is basically, I'm not going to tell you something's bothering, right? I'm very stoic, I'm quiet, I'm here, right? The really good example of this, and I think it was better than we are the way we are now, right? A stoic person does not share feelings very well, right? I'm not going to tell you this is bothering me. My wife's grandfather in World War II was stationed in the Philippines and was part of something called the Bataan Death March. If you want to know what that is, Google it. It's incredibly terrible. Okay, he came back from being in a prison in the Philippines for years, where they amputated his leg with, just like have a couple of shots of whiskey. Here we go, kinds of things on this because he had gangrene. Anyway. Never talked about it, ever. Never. Would not talk about it, would not speak about it. In some ways, that is an idea of stoicism, right? It's a very sort of this 1950s be a man, don't talk, right? It's a very traumatic experience. So we're looking at, there's some evidence about stoicism and pain as well. People that are stoic, it may hurt, but you won't tell us that it hurts, right? And so, is there stoicism in the midst of this? Is that stoicism related to childhood trauma? Is your anxiety and your catastrophizing related to childhood trauma? Maybe. I don't know. We're doing the same. So we would appreciate you guys putting the questionnaire if you want to. Okay. Let's talk very quickly about the least important things ever, which is pulmonary system. Are there adaptations in the pulmonary system in response to aerobic exercise? And the short answer is no. Okay, your lungs are your lungs. They're not going to get bigger. Okay, you don't get more alveoli, you have thinning now. Okay, what we see if we measure your breathing rate and breathing depth after training at rest, no differences. Ventilation is the same. During submaximal exercise at any given work rate, breathing is basically the same. Okay. As lactate threshold increases, your ventilatory threshold increases. Not a lot going on there. Where we do see changes is that people that have a higher VO2 max have a larger maximal ventilation. We don't think that's because something changes in your lungs. All of you could have a higher max ventilation right now, today, if I let you exercise at a higher thing. I give every one of you a pint of red blood cells right now. I make your VO2 max go up right now. Your ventilation goes up immediately. What that tells us is that ventilation is not limited. The lungs, as long as you have normal function, the lungs are rarely ever the problem that limits you from exercise. Even when you think, I can't catch my breath, I can't breathe deep enough, this is a problem, That's not the real issue. Okay? Now, if you have a restrictive or an obstructive pulmonary disorder, if you have emphysema, if you have cystic fibrosis, right, something like that, then yes, it absolutely can have an effect. But in otherwise healthy people, it's not gonna change. Okay, there's no real adaptation. You're just gonna be able to better target kind of your total capacity. I got like 2 minutes. We're gonna finish our adaptations by talking about cardiovascular system. The first cardiovascular adaptation that you probably don't ever think about, but it's really important, is when you train, you get more blood. Your total blood volume will go from, say, 4 or 5 liters to 5 or 6, or sometimes to 7. Okay, maybe not to 8 for most, but your blood volume goes up. You have more plasma and more red blood cells. Have more red blood cells, there's more oxygen in your blood. More oxygen to go through those capillaries, to go into those mitochondria. It's a very, very powerful adaptation. How does that happen? Hormones. I call this mechanism, and I don't know where I heard this, but I thought it was great. It's a persistent acute effect. You go run today, your blood volume will go up a little bit tomorrow because you release aldosterone and antidiuretic hormone, and you actually make a little bit less urine. Your plasma volume initially will start to rise. Okay, you may be a little bit thirstier, you may drink more fluid, but you do that, and it stays up for a couple of days. And then if you don't exercise again, it goes back down. You eat the plasma away. Okay? So exercise today, it lasts for a couple of days. If I exercise again in a couple of days, I get that same effect again. And again, and again, and again, and again. So it's an acute effect that lasts for maybe 48 to 72 hours. But if you are persistently exercising in that window 4 or 5 times a week, you're always in this sort of zone where you're stimulating that little bit of retention of extra water in the kidneys to expand your plasma. We'll talk about where the red cells come from. Okay, have a good weekend. Make good choices. If you really want to know about your test, you can send me an email and I'll look it up.